Welcome back. Welcome back, everyone, to the fifth day of AICTE sponsored Faculty Development Program, that is FTP. Once again, I am Sarfraz Anwar and I wish a very good evening to our honorable speaker, respected faculty members, and all the participants. I hope you have all joined us with the same energy and enthusiasm to learn and explore together once again today. So, first of all, let's take a quick look at today's schedule. From 6 to 7:30 p.m., we have a talk by one of our youngest and most dynamic speaker, Dr. Viswajit Mulpuru, and he will be speaking on concepts of next generation sequencing. And then from 7:30 to 9 p.m., we will have a talk by a very inspiring speaker, Professor Kunal Roy, and the topic is in silico toxicology using QRA, QRASAR, and ARK approaches. And I am requesting to my technical team that please confirm whether our honorable speaker Dr. Viswajit has joined us or not.
No, surprise, he has joined.
Okay, ma'am. And I am very happy to share that Dr. Viswajit Mulpuru has joined us today. So, before we begin his informative and insightful season, I would like to invite the co coordinator of this FTP program, one of our finest scientists and associate professor at IIIT Allahabad, Dr. Sintu Kumar Samanta. Sintu sir, we are requesting you please give a brief introduction and warmly welcome Dr. Viswajit with great enthusiasm. So I would now like to hand it over to Dr. Samanta sir.
Okay. Thank you, Sarfaroz. Am I audible?
Yes sir, you are audible.
Okay, fine. Um, Dr. Biswajit Mulpuru is a computational biologist with expertise in genomics, molecular diagnostic, and AI-driven bioinformatics. He brings deep experience in next-generation sequencing, variant analysis, and mutagenomic profiling. He has done his MTech and PhD from IIIT Allahabad under the supervision of Dr. Nidhi Misra during 2017 to 2022. Then he moved on to CCMB Hyderabad, where he joined as senior project associate. During February 2023 to May 2024, he worked as an associate professor at Vigyan's Foundation for Science, Technology and Research, Guntur, India. And currently, he is as the head of bioinformatics in IPACITY LLC. So, as this organization, he plays a key role in building scalable bioinformatics pipelines and data interpretation tools that power advanced diagnostic and research solutions. With this brief introduction, I extend a very warm welcome to you, Dr. Mulupu. So, over to you. You can then start the presentation now. Hello, Sarpros. Dr. Mulupu has joined, na?
Sir, Nidhi ma'am has informed me that he has joined.
Then let's wait, I think. Can't you see in that list of participant?
Sir, he is not. His name is not present in that participant.
Okay.
Hello, I'm audible.
Yes.
Yeah, yes, join. Okay.
Okay, just give me 1 minute. I'll just
Hello. Hello.
Yes, ma'am.
Vijit has joined.
Yes, ma'am. Yes, ma'am. He is joined.
Okay.
Hello. I hope my screen is visible.
Yes, sir. Yes, sir. Your screen is visible and your voice is also clear.
Yeah, thank you. Yeah, good evening, everyone. I'll be actually giving a small talk on the application of bioinformatics in NGS and pharmacogenomics. Yeah. Okay. So basically, here I'll be connecting two threads. See, basically, like everyone knows NGS, and then pharmacogenomics is actually a bit newer topic. So, over here, we'll actually see how NGS and pharmacogenomics is useful in clinical setting. So what happens in bioinformatics actually? So we actually turn raw sequence reads into some VCF files and all, right? But in a clinical setting, we actually have to turn this into a trustworthy, clinically usable variants. And second, these variants should also drive concurrent prescription choices, because that's what pharmacogenomics actually tells about. The main message is it has to be accurate, and the pipeline that we actually use should be completely reliable and should be reproducible. So I'll be taking you people through a pipeline steps so that we can generate a pipeline that is reliable and reproducible and even clinically relevant for clinicians and all. See, in a clinical setting, bioinformatics is not a exploratory hacking and all. In labs and all, we'll use some NGS pipelines and all, and we get some results and all. That's not what happens in clinicians, right? It has to be highly reliable, and it has to be version controlled. In the sense, you cannot use one particular version today and say, okay, I have used some other version tomorrow and all, because it has to be completely reproducible and all. And this mindset actually changes everything, right? Because we have to pin the references, we have to log the versions and record every single parameter that we use. And there has to be auditable trial, like that, and it has to explain the complete result and all. And over here, because I hope many of you are actually PhD students or faculty members and all. So there you will actually be judged by how novel you are. But in a clinical setting or in a industry setting, that's not what we want actually. You are not judged by the novelty. You are judged by the reproducibility, clarity, and the turnover time. That's a very important thing. The turnover time is one of the most important things that we have to consider. And in a clinical setting, we see a file, because in normal labs, okay, we'll be giving a FASTQ file, we'll be getting some VCF file, some interpretation and all. But that VCF file is not the deliverable over here, because over here clinicians should actually act. They have to act with confidence. So we have to standardize the intake. We have to give a complete interpretable result and all. And the pipelines that we use should be so reproducible that if you actually use the same result today and even some 6 months or 10 months or a year apart, you have to land on the same result, except for few additions like a database update or some other regulatory kinds of things that can be an exceptional. Yeah. Yes, I have previously told, clinicians don't want data, they want an answer. They want an answer in a very niche way. And our contribution is to compress this particular FASTQ file data into a single line or a single page report, basically. So we actually add value in 4 different ways in these things. In our company, we actually add values in multiple ways. We actually perform NGS analysis, we perform pharmacogenomic analysis, we perform AMR analysis and all. But I will be taking you through the NGS and as well as pharmacogenomic analysis over here. So why NGS? Basically, it tailors diagnosis that we all know, and it helps in personalized care and all. And it helps clinicians get an informed decision and improve outputs and all. Basically, a personalized medicine and all. Okay, so people use various platforms. It can be Illumina, it can be Nanopore, it can be whatever, some other company and all. But ultimately, the output is a FASTQ file over here. And what will be in a FASTQ file actually? The FASTQ file is basically a collection of a set of 4 different lines and all. The first line will have a label, the second line will have a sequence, and then we have a spacer known as plus, and then the most important, that is the PRED score or the quality score over here. So basically the sequence and the PRED score or the quality score is what helps us actually determine the variance over here. Okay, so we have a FASTQ file. So what do we do now? The first major thing is quality control. So why quality control? Because you see, like, sorry. You know, the samples are loaded by a laboratory assistant or some other person from a wet lab setting, right? So they might do some unavoidable, some mistakes or whatever, or maybe some reagent failures or whatever can happen. So quality control is the most important aspect that you have to consider. So how do we perform quality control? The first thing is there are various tools. The most important tool is we use FastP over here in our laboratory. And we perform it before alignment. So we verify the concentrations, the fragment sizes and all, and then we actually consider these particular scores, and basically these are in ASCII format, and each particular symbol over here represent a number from 0 to 99 actually. And in our quality control, we have to consider only those bases that are above 30 quality control number. That is instances per error. That's a difference error and all. So, and then, you know, when we are performing a sequencing, we add various adapters, we add various primers, adapters and all. We have to actually trim them. We have to remove those adapters and all. That's known as adapter contamination. So we'll be using the FastP again to remove the adapter contaminations and even check the DC bias, the duplicate rates and everything and all. And all the low quality regions will be removed over there. And then we'll be going for alignment. Okay, alignment is actually, there are various tools for this thing. Basically, we'll be taking a reference genome. We have a human reference genome. So we'll be taking that and use a aligner. And BWA is a major aligner that every clinician use, every clinical setting uses. But yeah, there are other aligners like BOTA2 and all. But BOTA2 is majorly for short reads, whereas BWA is for a long read alignment, you know. And then there is MinMap2, which is again another common things that we use. Okay, coming to reference. Again, I said it has to be consistent, right? So in references, I mean, for a human, there is a GRCS 37 reference. There's a GRCS 38 reference. Both are from the human project, you know. So if you're considering, second. Yeah. So if you are considering one particular reference in your pipeline, you have to continue with that pipeline, that reference in the for complete output of the pipeline. Yeah. And you have to actually even label them and give those references in the report and all. Okay. So alignment is done. Now next what? We have to sort and remove duplicates, because see, in sequencing, you perform basically it's a kind of PCR only, right? So there might be several other duplicate templates and all that might have happened over there. So we have to actually trim all of them or remove all of them so that we have a normalized genomic output and all. So we'll be using tools to remove the duplicates. Basically, we use GATK and PyCard to remove the sort and remove the duplicates over here. And yeah, there are tools like SAMtools also. And then comes the actual part over here, the variant calling. Okay, the quality control, alignment, and sorting, removing duplicates is a part of almost all NGS pipelines and all. But what happens after this is what is unique to the clinical setting and all. So preparation for variant calling. So what is actually variant calling? Okay, see, every single human will have their own genome. We all know that they'll have their own mutations and all. So when you actually see this particular genome, you will be getting n number of mutations. You will be getting because we are actually aligning it with the reference, right? So we'll be getting thousands or maybe a few lakhs mutations and all. But not all of these mutations are actually pathogenic or something, right? So we have to actually filter out these pathogenic variants over here. So how do we do that? So basically we use various filters. They are actually standardized filters for them. We use standardized filters to remove all of these unnecessary mutations and all, unnecessary variants. Okay, but anyway, before going to this filtering step, the most important step is base quality score recalibration, that is BQSR. See, as I've already told, we all know that there are various mutations, right, in the humans. But when you are actually considering this quality scores or removing the duplicates, removing the low quality reads and all, you might actually stumble upon various artifacts and all, right? So we have to remove those artifacts because they might actually cause some false positive results, false negative results and all. So we will be actually taking few known databases, and it can be DVSNP or database from a 1000 genome project. And majorly the gold standard is the Mills and 100G, 1000G gold standard indels, which is by the GATK and recommended by the GATK also. So we will be taking those indel database and compare our particular aligned genome to remove the artifacts and all, and even recalibrate the base qualities and all, because sometimes when those artifacts are present, your quality might be different. After this thing, the quality might be different and all, because we actually require a high confidence output, right? So we'll be actually using this databases to remove the artifacts over here. So what happens in BQSR actually? BQSR actually removes the sequencing errors. It penalizes the non-needed variants and all that. But yeah, because why are you might have asked, why are we actually giving this particular known sites? We are giving these known sites because we do not want to actually remove these particular sites, even if there are some mutations over there, because they are actually known variants over there. Even if there is a small amount of polymorphism over there, we do not want to remove it because that might be a somatic mutation. It might be a minor germline mutation, right? So that's the reason we actually use the databases to see, okay, this region might actually have some variant. So even if you find a minor deviation over here, do not remove them. So that will actually improve accuracy levels. And then, okay, we have performed BQSR and all. And again, like we will be having lakhs of variants again. So how do we actually filter out more? We will be using various filter parameters that range from depth, the quality, even the strand bias, and the mapping quality. And these all we'll be seeing about this thing in the few other slides. And we'll be using these filters to remove the high confidence, to only get those high confidence variants and all. So what is depth actually? Okay, see, in suppose in a real-time PCR and all, we'll be getting some CT values, right? Okay, we have a template, we have a primer and all, and then we see, okay, this particular variant is actually started amplifying at this particular cycle and all. Similarly, over here, we'll be seeing how many times this particular region of the genome has been amplified. Because we do not need the low amplified regions. Those are very low confidence and all. So we will be only taking very high confidence regions over here, and that is based on depth over here. So depth actually represents the number of times that particular region has been covered by the sequencer. So typically, in case of a germline, it has to be 20x, 30x sometimes, and a somatic, it has to be at least 30 to 50x, and sometimes we even go to 500x if we think there are some artifacts and all. And yeah, like sometimes you might get a very high depth. That may not be a somatic mutation. That might be due to a copy number variation. There are various genes that show copy number variations, right? So we have to even eliminate those and all. And then comes one of the most important aspect, that is known as strand bias. Okay, each DNA will have reverse and forward strand, we all know that. So what if only one strand has actually amplified? Then maybe like you might see, okay, this person only has homozygous variants, all of them. But maybe that person actually had a heterozygous variant over there. So how do we actually know that? How do we check if both the strands are actually amplified over here? So for that thing, we'll be using figure strand and strand dots bias, that is FS and SOR parameters, to remove the regions from the strands that are actually showing bias towards one particular strand and all. So the general cutoffs over here is for FS is greater than 60, and then for SOR is greater than 3. So if it's greater than 3, we'll just remove the particular region and all. So we'll be actually getting only the regions that had a perfect balance between both the strands and all. Because in case if we consider an unbiased sequence over there, we might actually see only variants from one particular region. Let's suppose a person had A by G, A on one allele and G on another allele. And if sequencer actually was biased towards only one strand, then we'll be considering that person as A by A and give us some other result and all. So that's a very unnecessary stuff that we have to remove and all. So that's the most important aspect that we have to consider over here. And then we have to go for genotype quality is another filtering technique. Okay, we have filtered based on strand bias, we have filtered based on depth and all. But what if that particular genotype was actually not very accurate? Maybe the genotype was A, but the sequencer had seen it as some T or some G or whatever. That's what we see over here, genotype quality. So we have to actually see, okay, if it's a G over here, it is actually G from the output, right? So we'll be considering genotype quality over here to remove all those low quality reads again, uncertain calls over here. So again, the typical threshold is greater than 30, but sometimes we even go to 99. 99 is the highest actually. So for a high quality clinical report, we mostly consider 99 as a threshold. But yeah, for a general germline and all, we can go for 30 and all. Okay, now we have all the high quality reads, the filtered high quality reads. But again, we know pseudogenes, right? Okay, there are various genomic regions with very high similarity, high repetitions and all. What if our sequence, our aligner actually done some mistake and aligned our particular reads into some other gene instead of some other gene? So that is where pseudogenes come into picture over here. So we have to remove all the pseudogene variants to remove the faults variants. So how do we remove them actually? Basically, we actually take a set of pseudogene annotations, set of pseudogene coordinates in the genome and map our particular alignment with that particular annotations and all. See if, okay, basically gen code actually gives us that particular annotations and all. But what happens over here is, okay, see, for example, we have PMS2, SIP2D6 and all. They have very high repetitive sequence over there. So suppose our sequencer actually mapped a particular genome onto some gene, but this particular annotations actually specify, okay, this particular genome mapping over here might actually cause due to pseudogenes and all. I mean, we had some annotations into that thing. That particular region is highly repetitive and all. So we will actually flag those as pseudogenes and actually cross-check them before going for next step. Okay, now we have removed all the unnecessary low quality variants. Now we have all the mutations of that particular patient or that particular person. But now we have to actually tell the clinician what are the pathogenic variants from that particular person, because that person might have hundreds or thousands of mutations again, right? What are the pathogenic variants of that particular person? That's what we have to tell the clinician, because that's what helps them make the decisions, right? So there are various tools again for this thing, and the major tool that we use is VEP, variant effectors and all, and there are various other tools also like star. Yeah. So basically, what happens over here? Again, there are a set of databases over here that we actually compare our particular output with and see, okay, if that particular variants already defined in the database as a pathogenic, then we'll say, okay, this particular variant might be pathogenic over here. And suppose if that particular pathogenic variant or some variant is not defined in the database at all, then we'll not even know what's that thing, right? So then we'll be defining that, we'll be giving that particular variant some other criteria based on the predictive scores and all, basically. But is there a standard way to classify them? Yes, there is a standard way actually. ACMG variant classification is something that we use over here. ACMG is American Council for Medical Genetics and all. They have a variant classification guidelines that every single laboratory actually have to follow and all. So what are these ACMG guidelines actually? As I previously told, this particular ACMG guidelines actually consider data from previously known databases, population data, some computational predictive data, and even evidence-based data from databases like CleanVar, from NCBI, from DBSMP and all. They collect all this data and specify each variant into various tiers and all. Basically, there are 5 tiers over here, and broadly classified into pathogenic and benign. And this actually helps keep the reporting as consistent and reproducible between laboratories actually, because even yeah. Okay. Okay. Although they are classified into pathogenic and benign over here, they are again subcategorized into subclassifications. Basically, there is a PVS1, PS1, 2P. There's a very strong, moderate, supporting benign, and all of them are actually subclassified into PS1 or PM1 and all. We will be seeing about them in next slides. So how are they actually classified? As I was told, we'll be considering population data, right? So population data is basically allelic frequency data that we take from Genome, that's a database, and 1000 Genome Project, and various other databases and all. So suppose a particular variant is a very common in the population and all, then we'll say, okay, it's a likely benign now. No, it's a benign variant, you know. And again, like our population in the sense, okay, suppose we have a sample from Asian and like some Asian or some Hispanic or whatever, some American and all. We have to actually take allelic frequencies only from that particular population, because they are the again variants between the populations, right? So one population might have one kind of pattern in the variant, some other population might have some other kind of pattern in the variants. And all right, so we have to actually consider the patient's origin also into when considering this particular interpretation now, because you know they are found variants and all, like basically in Jews actually there are many different variants that are actually not present in many other races and all. Yeah. So that's an example over there. Yeah. And again, we will be actually considering various predictive information also, and most of the tools that we use are SIFT, Polyphen, and CAD that actually predict the pathogenicity of that particular variant. Is it a pathogenic, or it's a likely pathogenic, or a benign variant? So we're using computational variants and even those tools. So okay, this particular variant actually could give a loss of function to the protein or a altered function to the protein and all. So even we'll be considering those particular outputs for the considerations and all. So that's the functional data. And again, yeah, suppose there's a variant that a person had and it was never reported anywhere. That's known as a de novo occurrence. That has never been seen in any other patient. In the sense that was not reported in the database and all. So that's considered something. Like there's another criteria for that thing. We'll be considering all this particular knowledges to give the ACMG guidelines and all. Okay, so what are these ACMG classifications actually represent? The very important thing is PMSV. That's a very strong. If we actually predict that particular variant shows a loss of function in a person, then we have to consider that as a PMSV or a very strong pathogenic variant, because that is actually responsible for a disease mechanism, right? Few examples are BRCA1 hereditary breast cancer, and then we have a frame shift variants of CFTR or a exon deletion of SMN1, and there is very other examples over there that actually fall under the very strong pathogenic variants and all. And then the second most tier is the strong, and this particular strong is actually reclassified into 4 different variants again, like tiers again, PMS1 to PMS4, whereas PMS1 is basically related to the pathogenic variant present in that particular person, but this particular pathogenic variant is caused due to a kind of a different nucleotide change, because you know, like they are 3 nucleotides for a amino acid, right? Maybe a different nucleotide has been changed and which causes the same amino acid itself and all. So that will fall under PMS1. And then PMS2 is a de novo variant. That I mean, like the variants that we have not previously seen and all will be classified into PMS2. And then PMS3 is basically those variants that have functional studies, and those functional studies actually show that particular variant is a pathogenic and all. And PMS4 is something where, okay, there is a variant, and that variant is actually present in the population and all. I mean, like it is present in the population, but suppose that particular variant is present in this particular person in a very high frequency, then it has to be present in that particular population. Suppose there is a particular variant that is present, the frequency is suppose 10% of the times in the genome and all in that particular population. Suppose this particular person had it 60% of the times or 70% of the times, then we'll say, okay, this particular person had this particular population variant, but in a higher frequency. So we'll say it will fall under PMS4 and all. And there are then moderate classifications, supporting classifications, benign classifications. Basically, these are we do report this, but not very relevant for the clinicians. Yeah, in the short term and all. Yeah. Yeah. Again, like okay, we are reporting all these variants, all these pathogenic variants and all, but all of this should actually comply with the regulatory standards and all. So there are majorly two different regulatory authorities, that's CAP and CLIA certifications, and that's very mandatory for each laboratory that's actually reporting basically in the US or whatever, in the other countries and all. And these ACMG guidelines also consider these particular certifications and set these guidelines and all. So majorly, if you actually follow these ACMG guidelines, you most probably be certified with CAP. But there are various other parameters that have to be considered, like the reagents that we use and various other things. Yeah. And this is one of the CAP report of our sister institute, sister laboratory, that we have passed all of the blind samples that were sent to us. Basically, we had 100% concordance with the CAP results and all. So that's about just basic NGS and basic variant analysis. So coming to pharmacogenomics. So what is pharmacogenomics actually? So until now in NGS, we have seen, okay, this particular patient or this particular person has this particular mutations, and this might cause some diseases. This might or might not cause some disease in future, or if it is somatic variant and all, okay, this particular patient had this kind of somatic variants and all and all. But in pharmacogenomics, we actually integrate this particular NGS variants into drug response. We'll actually tell the patient, or actually basically the clinician, does this particular genotype, this particular patient or a person will be translated into this particular phenotype. And this particular drug might show a low metabolism, higher metabolism, or no response at all in this particular person and all. And we'll be actually giving a alternative approach to them, to the alternative drugs, suggest alternative drugs to the clinicians and over here. So what are the major pharmacogenes? Because there are n number of genes in the humans, but there are only few pharmacogenes in the humans. And the major genes are actually CYP2, D6, all the CYP genes actually, CYP D6, CYP2, C19, CYP2, C9, CYP2A, 5, whatever. There are multiple pharmacogenes, and CYP2, D6, TPMT, and SLCO1B1 are the major genes that actually influence the drug metabolism. And we know CYP genes are actually part of the liver metabolism of the drugs, right? So we will actually consider the variants present in this particular pharmacogenes to suggest the clinicians, okay, because this particular variant is available in this particular person, do not suggest this class of drugs, suggest some other class of drugs to this particular person. So we'll actually help them predict the adverse reactions of the drug. So how do we actually do that? See, as I previously told, there are a very small set of genes that actually are important in the clinical setting and all. There are CYP2C19, D6, C9, and A5. Okay, basically these influence the activation because when we consume the drug, it has to get activated, right? And basically these four particular genes actually influence activation, even the clearance of the drug by the liver, the kidneys, and all. And then we do have TPMT and UDT15, which actually impact the metabolism of a drug, majorly theopurin, and the toxicity risks that a drug causes and all. And then we do have UGT1A1 that actually influences how a drug is handled by the bilirubin and all in the things. So how do we actually get them? So there is something known as star allele definition over here, star alleles, and that is defined by the haplotypes. So what are actually haplotypes? Presence of a set variants in a person, or a presence of a set or a set variants is known as a haplotype. Suppose he had suppose there's a variant at a chromosome 2 at this particular location, at a location some 100, and there is another mutation at a variant 150 and all. And suppose these two actually show some interlinking metabolic activity and all, then we'll say, okay, this particular variant A and B actually commonly known as a haplotype and all. And these basic haplotypes actually define the metabolic state of drugs they metabolize. And then there is copy number variation. Suppose majorly CYP2D6 and CYP2C19, they have copy numbers variations and all. Although they are the genes that have copy CNVs, these are the majorly clinically relevant variants. So suppose there is a drug that is metabolized by CYP2D6, and general population will have two copy numbers of it, right, on the first strand and the second strand. Suppose a person had a third copy number of it, then that particular person will show a rapid metabolism to the drugs that are metabolized by CYP2D6. So then in that case we have to alter the dosage and all. So that's how we use the CNVs over here. Okay, this is a basic example, brief example of what star alleles over here. See, suppose you can see over here there is some variant, some RSID, some variant at a deletion of T at this particular location. So when there's a deletion of T at this particular location, then we can say, okay, there is this particular person had a *3 star allele for CYP2D6. And because he had a *3 star allele, then he'll have no function of CYP2D6. He does not have any functionality of CYP2D6 in his organs. Or maybe a person had this particular haplotype over here. You can see over here we have an example of 3 variants over here. And with this combination, he'll be having a star level of star 11. Again, star level actually represents again a no function and all. And you can see that's interesting aspect over here, star 11 and star 14. Majorly, two of these variants are very actually identical, and there is one variant that is actually different between these two. And this particular change in variant actually represents star 11 or star 14. And when they have a star 14, it's a decreased metabolism instead of just a complete loss of function. And again, they are again standard variants defined by CPIC. Again, CPIC is a regulatory body that defines this the effect of each particular variant on a human body and all. And that's again a evidence-based approach over there that they consider. And what are the various tools and databases for PGX? The major tools that we use for a person uses actually FarmCat, which is actually a kind of a standard tool. FarmCat is a standard tool that we use. And then we have PyPGX, Stargazer that actually help us get the allele calling from the NGS and PCR data and all. And then we do have databases like FarmGKB, which actually curates the gene drug associations, which particular gene actually metabolize which particular drugs and all. So we'll be actually taking all these particular databases tools into consideration before reporting the NGS samples and all. And there's a brief example of a CPIC guidelines over here. See, suppose they actually update these particular guidelines very often, and we have to keep updating the particular pipelines based on these guidelines. You can see there's an example of a CYP2D6 where suppose a person had a star 1 and star 1. It would be basically a normal metabolism. But over here, he had a copy number of M. We had a copy number of, although it's a star 1 by star 1 is considered as normal actually. So although he had a star 1 by star 1, he had an extra copy number over here. So we'll be considering it as a ultra rapid metabolism over here. And then we have various other examples of diplotypes. Here a person had a star 1 by star 4, or star 1 star 5, and various other parameters. And based on these guidelines, we'll be modifying our particular pipelines. And again, this is another example of CYP2C9 actually. This is another CYP gene, another pharmacogene. And the guidelines and all. Okay, there are a few case studies in PGX actually. Okay, so we are actually getting this particular star alleles and all. So how are they actually being used in the clinical setting and all? See, suppose a patient had a CYP2C19 and a variant was star 2 by star 2. Basically, star 2 by star 2 represents poor metabolism. Because this particular, for a particular drug and all, suppose the doctor was prescribing this few particular drugs, Copilogrel over here, and suppose that star 2 by star 2 is actually represent a poor metabolism for that particular drug. Then the doctor or clinician will actually give a alternative medication to that particular person over here. And then we have TPMT variants over here, where basically TPMT actually influences the therapeutic metabolism. So if there is a genetic variant where there is a rapid metabolism of TPM by the TPMT or a smaller metabolism, then they have to adjust the dosage based on that thing to avoid toxicity and all. This actually helps in personalized treatment and all. Yeah, the coming to challenges actually. okay, we have the data, we have the interpretation, we have all the databases, we are following the guidelines and all. But what are the challenges in implementing this? See, as we have previously seen, considering the PGEC itself, there are multiple variants that we have to consider for haplotype. And again, suppose a laboratory has not been considering NGS over here. Maybe they are only going for genotyping based on PCR, RT-PCR, genotyping of few particular variants. Suppose I'll just go back a few slides over here. Yeah. Suppose there is a laboratory which was considering genotyping based on PCR, and they are only having templates for few variants. Suppose they have only had variants for these three variants, the star 11 variants, and not for this particular variant, the last variant that we see in the star 14. So suppose, so in that case, we'll be getting a genotype based on that thing, right? We will not even see this particular variant over there. So what we actually give to the patient might not be completely true because we haven't even received that particular data over there, right? So that's one limitation over there. But in case of NGS, that's not a majorly a issue because we'll be considering the whole genome sequencing over there. So interpretation of this complex variance is the major limitation over here. And then data privacy is actually, again, a very mandatory aspect that you have to consider because every single data that we receive, every single patient's information, it can be genetic information, it can be their personal information, everything has to be securely saved and all. And then there's cost and resource demands, and actually, because establishing a lab is actually a very costly process, right? We have to have the sequencers, we have to have the templates, we have to have the manpower and all, the technical resources and all. And again, the most important thing is EHR integration. Because see, suppose a patient has, or a person has come to us, to a laboratory, and they have performed PGX analysis. And they have known, okay, they are not, or they are a poor metabolizer for some class of drugs. And now they go to a doctor and all. And he'll actually prescribe some kind of drug over there. So now how do we actually link this particular PGX report and send it to the doctor and all? So that's the most important aspect that we have to consider. We have to integrate the doctor's portal and this particular laboratory portal so that it's a seamless integration between, and the data transfer is actually reliable. And again, we have to consider the data privacy into consideration over here. And what are the emerging trends that we are actually seeing in this particular pharmacogenomics or clinical settings of NGS and all? Basically, they are increasing usage of AI in various interpretation. And again, like, although we have been using AIs, it has to be very judiciously used because AI is not always right. There has to be a manual interpretation in between and all. And then there's another thing, real-time sequencing, because right now all the sequencing is done in the laboratory, and then we receive the data, and that data will be in few GBs, sometimes even in few TBs and all, and that data transfer takes some time, and then we have to run the samples, then we have to generate the reports and send it back to the lab and all. So real-time sequencing actually, and data collection at the source itself, will actually help us decrease the turnover time between the sample collection and report generation and all. And again, multi-omics approach and all, again, because you see, like, over here itself, we have seen two particular variants, right? NGS and PGX. suppose a person had gone for NGS at some other time and the PGX at some other time, we have to integrate both of them, and then we have to give a comprehensive report to the clinician and all right. So that's again a challenging approach, because both of the reports actually matter to the clinician for treatment and all. Yeah. And again, like new clinical utilities actually help in establishing the relations and all. So that's about the basic understanding of what happens in NGS and PGX, and what are the pipelines that we use, and how is the flow of the data between the laboratory and the particular report generation and all. I hope it has been a bit clear about the pharmacogenomics and next-gen sequencing. You know. If you had any questions related to this, yeah, again, I'm very happy to.
Thank you so much, Dr. Viswajit, for that wonderful and informative season. And now we will begin the question and answer season. And dear participants, I have a very kind message for you all. Don't be shy. Even the best scientists started by asking questions. So please feel free to share your queries or thoughts. There are some questions in the chat box. So I am reading that question. The question one is asked by Rameshwar Sughanti sir. The question is, how will your FDP on annotation and regulatory benefit rural India, where healthcare infrastructure and awareness are limited, and what improvement can be expected in their health outcomes?
Yes, so basically, because I have previously told, we do have to integrate our particular patient data, the diagnostic lab data, and everything, and actually have a specific portal for all of them like that. But that's a very long job for us to do. So we have to actually create the awareness with doctors. We have to create the awareness with patients that these are the tests that we have. What are the benefits of this particular variant analysis? What are the benefits of this pharmacogenomic analysis, and how they actually affect their clinical treatments and all? And that's a thing that we have to actually consider into picture before actually getting this into the Indian environment or any environment, because the knowledge of these particular reports to the clinicians and as well as the patients is actually what is that thing that we have to consider before taking these into mainstream and all.
Okay, there is one more question from Rameshwar sir, and that is what challenges may arise while implementing the annotation and regulatory strategies from your FTP in rural India, and what strategy will your team adopt to overcome that?
Yeah, can you please repeat the question once?
what challenges may arise while implementing the annotation and regulatory strategies from your FDP in rural India? And what strategy will your team adopt to overcome them?
Yeah, basically the first thing is all the regulatory uh, guidelines that we consider in our pipelines are from, it can be from CAP, it can be from CLIA, it can be from CPIC, and majorly all of these are regulatory bodies from the United States. Uh, and they are, I mean, like as per my knowledge, there are no major guidelines in Indian context on these particular variants, although they are like NI, NBA accreditations and all, but there are no major guidelines on these particular reports in Indian context. So the first thing is we do require a few guidelines on these things.
I think Rameshwar sir would be satisfied with your answer. And there is one question from Lovely Rat, ma'am. And the question is, what are the 5 rights of CDS and how do they apply to a PGX alert in an EHR?
Fine.
What are the 5 rights, RIGSTS, rights of CDS, and how do they apply to a PGX alert in an EHR? You can see the question in the chat box
I'll
also
just see the
if you have any problem.
Yeah. Right. Yeah. Basically, like I don't understand, but can you please elaborate the CDS?
Hello.
There is
Miss Lovely Rath, can you elaborate? What do you mean by CDS?
oh, okay. There is one more question. What tools we will use to secure PGX data which we are using is current and regularly updated?
Oh, basically we use industry grade servers over here to have all the data in our servers. It can be like basically everything is encrypted in our database, and only prescribed or our partner clients can actually see them based on the encrypted, using the encrypted keys. It can be password, it can be another walls and all that. So everything is actually encrypted in our servers, and it generally do not leak outside of the labs and all.
Sir, so basically we are not using any tools regarding our current data security. I want to know any tools are there, if any.
Yeah. They are, I mean, like, because see, they are data security everywhere, right? It's not just in healthcare. It's data security in banking system and all. Banking system, various other, even you have data security in email login and all. So every particular data that we actually get is secured in our servers, which is actually managed by our networking security team and all. And only the laboratories and our particular clients have the access to them, and then do have the very stringent security guidelines over here.
My first question was, sir, actually CDS means clinical decisions support.
Okay. Basically, because we give complete evidence-based results over here. Everything is reproducible. As I told you in the starting itself, everything has to be reproducible. And just because uh, it's a like a just like a PSD, like laboratory and all. It has should not be some exploratory things and all. It has to be completely reproducible. We will be logging every single parameter, every single tool, every single version of the tool, and the complete logs will be saved over there. And suppose even if you run this particular sample even after 6 months and all, we'll be getting the same output and all, except except we actually update some databases like because regulatory bodies sometimes update the guidelines and all that. In that case, we will be actually updating our database also. So only in that case, we'll be getting some changes. Otherwise, it's a completely evidence-based approach. So so the information to the patient, to the clinician, everything is transparent over here.
Thank you so much, sir.
Is there any other questions? Participants, you can ask. Don't hesitate. It's a very big opportunity to clear your doubts, to share your ideas. Any question from the participants? Looks like everyone understood the talk so well that there are no questions left to ask. Please ask any questions. Okay, I think there are no any questions. So thank you so much, Dr. Viswajit Mulpuru, for your wonderful and insightful talk on the concepts of next generation sequencing. You explained it so clearly that I think even our genes are feeling well sequenced now. So we truly appreciate your time and effort in sharing such a valuable knowledge with us. Thank you so much.
Yeah, thank you.
I think there is many some time left in the starting of the next season. So
It
currently
is
it is
around 20 minutes there. So we can take a break, I think.
yeah, ma'am. So for the participant, there is a small break, and you can take a short break. And please free to grab a cup of tea or coffee and refresh yourselves. So we can all come back energized and ready for the next exciting talk. So now we will again meet at 7:30 p.m.